Hey everyone, I just wanted to spend a moment talking a little bit about our experience today out in the woods and making sure you felt comfortable for the brief homework assignment that we're going to have associated with it. So great work this week. And I'm going to orient you to some materials that we've posted. All right, so Sandra Cook out of Greensboro, North Carolina, where her college is, put together this overview page, and I wanted to make sure that we spend some time on it together. So again, her interest is in monitoring these sapsuckers. And you can see there's a whole diverse group of sapsuckers whose behaviors are quite similar in terms of the way that they address trees and feeding on the trees, lapping up their sap using their unique tongues. We have, as I mentioned, the yellow-bellied sapsucker. And as you'll notice, it's really the only one that we would encounter in our region. And you are all familiar with what their interesting grid-like sap wells look like. And do they harm the tree? So there's probably, some questions as to that, whether or not that is the case. And in some cases where there's a lot of them, there may be a situation where disease can creep in, a fungus could build up, a bacterial infection, just like, in humans. And so similar to what we discussed with galls, you can imagine that perhaps, down the line there could be, and there's likely maybe some research out there that discusses that. In most cases, though, her literature searching suggests that there isn't, and that it actually might be a benefit to the community. And so that's where this kind of plays into the discussions that we're having this week. So we're going to be talking about keystone species. We've mentioned them already, but these are organisms that are not super abundant in the community, but as a result of their activity, their behavior, their niche, their role in that community, they are bringing in and allowing for other species to flourish. So hummingbirds, warblers, red squirrels, chipmunks, wasps, ants, butterflies, all profiting from the holes, the little excavation, and the leaking, sugar water that they're leaving behind as a result of that activity. So we can deem them a keystone species, and it's interesting to note that she actually even talks about them maybe being double keystone species, because not only does their feeding behavior increase the number of species, but their cavities, when they dig deeper cavities, they can result in a profiting other species. For example, flying squirrels might use those cavities. So double keystone species, perhaps. Ecosystem engineers, they change the habitat, add habitat complexity to it, and as a result can enhance the diversity in that region. So what we're interested in is kind of figuring out, what are their habits? What trees do they seem to forage on the most? I mean, we were in a floodplain, so we have limited, tree species that were located there. But if we were out in the main Ruger woods, we might see that they're using, different species of trees. Maybe they're using the birch more often, whereas we had a lot of, cherry and some cottonwood, some, northern white cedar, as well as some white pine, and you guys also had some ash. And so we're kind of limited in the diversity of trees that we had in our particular location. So, does the tree species matter? Certainly there's different barks, there's different sugar qualities. Does the orientation in which they're feeding matter? There's wind that could kind of cause some problems, wind exposure. Thermoregulatory issues, right? Their tree size may matter, and also just human activity could matter as well. So, what we typically see is that, sapsuckers are known to feed on a whole bunch of different types of trees, especially the one that we have in our area. They generally prefer maples and birches, something to consider, as we move forward. And evaluate things, but they can also forage on other trees. And I should say, what I learned is that they will often switch over to hemlocks and more of the softwood species when there are drought conditions. They tend to maybe retain moisture. And the conditions are a little bit better when it gets to be a bit more dry. In terms of orientation, there are studies that suggest, where the longer length here is usually the place that they prefer to feed. So the directionality that they tend to feed appears to sort of be in the southwest direction, definitely westerly orientation. So it'll be interesting to see if that's what you guys found. And in terms of the trees, interestingly enough, and maybe anecdotally you notice this too, they tend to go after maybe some of the bigger trees. And in terms of the bark texture, it looks like perhaps the thinner, less complex bark are ones that, certain researchers have found that they might be attracted to. And, thinking about our human influence, did we see a lot of sapsucker wells along the edge of a trail or along the edge of the river, or right in areas that seemed a bit more disturbed? That seems to be a trend that researchers have noted. Seems like they also perhaps will target some of the trees that are already damaged themselves. And perhaps are in a little bit of distress as well. So in terms of what we want to focus on, we're really curious about the distribution of these wells within the community. There's been some research in other areas, not much going on in our area though, so very important for you guys to get involved in this citizen science project. And we're interested in being able to scale this up, as I mentioned, right? We are collecting at the local scale here in Plattsburgh and in Peru and in this region, Clinton County. If we've got other New York colleges that are working on this question, they can also contribute, and we'll have regional data for New York, and then we can hopefully get to the continental scale, which is where the NEON complex comes in. So again, these are the National Ecological Observatory networks. There's research ongoing long-term in all these different locations. They've got sensors, they've got towers that have telemetry looking at temperature and other abiotics, pollution, moisture. And in addition, there's researchers that go out and do small mammal trapping and lichen surveys and a whole bunch of different things. So we could tap into, data to learn more about what the regional context is, with what are the conditions in these areas, and then pair that with the research that you guys are doing at the organismal level. The project is run through ERIN, the Ecological Researchers Education Network. Remember, I had mentioned that we are one participating college, but there's all these other colleges across the country that are participating in these ERIN projects. And so we'll have ones that we can compare our sites to. So our question is, how does urbanization affect sap well occurrence and abundance? And we might be kind of curious to consider how these trends might shift according to the NEON region that we're looking at. So again, we're in the Northeast region, maybe it varies. Do we see a directionality in terms of prevailing winds, light conditions, those abiotic stressors, right? Perhaps the birds are kind of trying to minimize that by picking a certain direction. And do we see any kind of, density distribution, so on and so forth? How does it correspond to those other species that we might find benefiting from them? And does it differ among, tree species that we find in our forest, or among native or non-native? Again, I think most of ours are native species, so we're not going to really perhaps be able to test that at this point. And so, what are we going to be looking at? Well, we're going to broadly kind of consider the continental scale, right? We're going to ask, what NEON ecoregion are we in? We mentioned the Northeast. Get your location. We're doing that with the smartphone and iNaturalist. You're determining your level of urbanization by coding it as urban or suburban or rural or wild. We're going to then move on and evaluate the Bortle scale. So the Bortle scale is going to give us the measure of darkness in our area. Again, just building on that urbanization front. And then we're going to get down to the organismal level. We're going to see sort of exactly, at the stand level, what are these woodpecker selecting for? So we're going to take all these measurements on our iPhone or using the data sheet and then adding them into the iNaturalist project later on. And And these are just some visuals of, measuring the DBH, whether or not the DBH, whether or not the tree is on a slope, how do you evaluate it? You get to the higher end and do the DBH from the higher end. Determining the species of trees. I mentioned Seek can be helpful. There's some other apps out there that are helpful. iNaturalist tries to ID. You guys also had visual laminated cards that would help for that. And bark characteristics, right? Important for us to scale that on a scale of 1 to 9, as we mentioned here, right? Based on the ridging and the whether or not they're horizontal or it's smooth or it's peeling. Is there, visual presence of disease? Do we see stippling or browning? Do we see some crown dieback? Do we see beech bark disease? Do we also see other tree cavities that might suggest that we have other woodpeckers in the area? And so all of the questions that we're addressing in the app will help at getting at these questions. We want to make sure that we evaluate the distribution at those different levels. And we want to note the abundance on a scale of 1 to 4 with binned amounts, and do that at all those 3 levels as well. And we're going to be walking around the tree to make sure that we have encountered that, right? We can see some furrows here, so this would be noted as an F perhaps. These would be noted as an H. Again, furrowing. We didn't see much of that. Holes, furrows, both. Again, doing that at all 3 zones. Orientation, none. If there's no orientation, leave it blank if no sapwells. All right, all this is going to go into iNaturalist. And if we have any further questions, we can reach out to Dr. Sandra Cook. So she's part of that Erin network who came up with this project. And as I mentioned, we are beta testing this. So very important for us to give her feedback on how the process went. I know we had some issues with certain phones. There's so many models of phones, so there's oftentimes some glitchiness and so on. But anyways, I think it's important for you guys to participate in this. This is legit research. It's awesome that you're, contributing the first ever observations. And to put it all in context, as I mentioned, we're dealing with a keystone species, which is something we've been covering in class. And we've talked a little bit about their importance in structuring and maintaining biodiversity in the community. They will often knock out that competitive dominant if they're a predator, leaving greater diversity in the community. In this case, they're kind of along the lines of an ecosystem engineer, in that what they're doing, they're fulfilling their niche. They're actually attracting other organisms, supporting those other organisms. And so the overview presentation that I just gave you, if it's useful in addressing your homework assignment, you guys can click it here. Again, it was very important that you guys do the pre-test for Sandra, Dr. Cook, and we're going to do a post-test afterwards to see how well you have addressed the learning goals. This is just an overview of our activity. And I embedded a video of Samuel and Owen and Andrew, and they are demonstrating the process of observing, making an observation for those, again, who were not able to attend today. Unfortunately, the one that they demonstrated had no sap wells, but I'm actually going to place in that same location, I'm going to place a screencast of what it would look like on my phone as I was making the observation. So you'll be able to see that in action. Here's some pictures of us in action out in the woods today. We were out behind the dorms and logging. We had a really good one here. We saw some skunk cabbage, which was pretty exciting. And here we go. If you go to iNaturalist and you go to the Sapsucker Wells on Trees project, you will notice, this is as of maybe 20 minutes ago, there are 42 observations, and I'm sure some of you are still adding those in. 12 species of trees were noted and 12 people participated in the project so far. And you'll notice it's all in the zone where we did our work. And if we were to, you know, open this up bigger, we'd probably be able to see on the map. This is a lot of the spots where we were doing our surveys. Might be some some were up on the slope. Could be some GPS error as well in there. And there's ones that have questions. They haven't yet to be confirmed with identification of species. So you guys might be receiving some emails from some of the quality control folks who have decided that they think it's this particular species, and you may just, you know, get a prompt to go in there and just approve it. Feel free to do that. But some exciting stuff. You can see, you know, you can find your observation in there if it's logged appropriately. So I posted that. And now I wanted to just briefly discuss the homework. And so you guys can click right here, and the assignment is going to be located there. The post test, you can do that after we've completed everything. It's the same series of questions. You may need this distribution map as you're doing this assignment. Okay, so let me orient you to the assignment. And here it is. So when you click on that, you're going to notice there's just a handful of questions, not much. Trees are considered a foundational species, just like the sapsucker is a keystone or a double keystone. Compare and contrast those terms. So you can find some of those terms in our PowerPoints, but also in the content of this module. What are the distributional trends of co-occurring species like the ruby-throated hummingbird, paper birch, which is again a common, and again for my lab today, we didn't see many paper birch down where we were. We saw ash. Paper birch is one of their preferred tree species. So we've made distribution maps of these common species that benefit from having the sapsucker. So that particular hummingbird, the paper birch, if you click on this map, it's going to bring up the location in iNaturalist, and I brought it into ArcGIS so that you can see sort of what the land cover is behind these areas. So this question is asking you, you know, what's the distributional trend and co-occurrence? And I suggest that you click on that and pull up the map. And when you do click on that, control click, you're going to get my little overview map. Okay, so what we have here is a map of the state of New York. So the darker green here is the Adirondacks. This is the Champlain Valley. In the yellow, more agriculture. This is the Tug Hill Plateau, Lake Champlain for orientation, Long Island. These are New York sapsuckers in yellow-bellied sapsucker. I made them yellow. Paper birch tends to be white, and the bark peels off. I made those white. And the ruby-throated hummingbird, I made those red. So you can see the distributions of them that came from iNaturalist observations. So people like you making these observations. And so we're asking questions about, you know, overlap and habitat use. You can zoom in on these and address those questions. I zoomed in on a couple of spots, and you guys can kind of evaluate. This is an Adirondack Park hotspot. This was New York State full extent, so that's all of New York. Here's the Adirondacks. Where do we tend to find them? This is the Hudson River corridor. Okay, where do we tend to find them? What sort of habitat? Are they overlapping? And here we have it, Long Island. Okay, are you seeing any trends in the habitat use or overlap? All right, so that will help you to answer that question. What's the most common characteristic of sap wells? What cardinal direction? You know, think back to your experience. You can go on to iNaturalist and you can download the project contents. So I'll show you how you can do that. You go into your project, sapsucker wells on trees. You can go over here to export observations as a CSV. Now, if you have entered data, you have to have entered data into the project, okay? Then you can come down here, and I like to, right before the create export, I like to hit all, create export, and in a second it's going to resolve, and it's going to give me a zipped file that I can download right there, and I can save it on my desktop, and I can call it oops, doesn't like those. And then you need to right click and unzip it. So I've just opened it up, right click, extract it. So there should be a folder right there, and here it is. I'm going to just rename it. My goodness. And then when you open it, you'll be able to see all the data that you imported. I'll post this for you guys. All right, so there's a lot of stuff that iNaturalist collects off your phone, okay? It collects a unique ID. We talked about this with the squirrels. Time, your user ID, you might recognize some of your code names here, right? It's going to give you a URL to the observation, a URL like you guys are familiar with, right? From the squirrels. Oh, look at that. And it's going to scroll over, give you your latitude and longitude, and then over to the right, it will give you the answers to those additional questions. So there should be a column here that has, you know, your native trees, your habitat, and then if we look over here, this is orientation, right? So you can kind of scroll through, see if there are any specific trends that you're noting about, you know, the directionality that you've got. Okay, so there's already, you know, quality control is something to consider. There's already a couple of things that you might want to perhaps go back and evaluate. This 833 is not one of the bark type options. So, you know, we might want to consult with somebody on that. But anyways, that will help you address that question on directionality. All right, so on which trees are they most commonly observed? Is there something unique about those trees that might influence their choice? You know, we looked at bark type, right? We scaled it on a 1 to 9. We looked at whether it also had other woodpecker damage or if it had disease, right? That might influence. And then make a prediction for the sapsuckers and allies as we face things like climate change, invasive species, you know, and so on and so forth. What do you think is going to happen to them? What's the trend, increasing, decreasing, and why? And then finally, I want you to sketch a community diagram, which is kind of cool. There's this program here, View, but you could easily do this in, you know, PowerPoint, where you could, you know, clip some images of organisms. You know, you might have the tree in the center, you might have your sapsucker around it, you might have some of those other species that we talked about. So think about the community in which the sapsucker plays a part, and draw this kind of diagram, this community web with arrows. You know, maybe you have a plus sign next to it if it benefits, the relationship is a beneficial one. Maybe you have a negative if maybe there's a raptor or, you know, fox that might eat them, you know. So feel free to kind of code them if you want to. The program View is is a program that actually helps you make these if you want to try that out. There's another program that might be useful to you, and I think it's on what I've sent to you in the handout or posted there. It was, I think, Philopic, which is actually pretty cool. So you can get the images, the silhouettes here. You can search for, see if they have one for Sapsucker. Sometimes they don't have them, but they might have one for like woodpecker. Oh, they do. So here we have a Sapsucker. And you can view the picture, and you can paste that picture, that silhouette, into a PowerPoint slide. Right, thumbnail, small, large, but you can copy that and place it into you know, your PowerPoint slide to make your diagram, or into view to make your diagram, because it'll be an image. And you can also do that for any of the other species. Again, they don't have like a ton, you know, they may not have every single species that we've got out there, but they do have a good bunch of them, and they're these really cool kind of silhouettes. So that's Philopic, okay, or Philopic. And feel free to use that as well. Okay. So have fun. That's the assignment. Not hopefully too difficult for you guys to process. It does involve kind of thinking broadly about the concept, the role of this organism in the community, and some of the data that you guys did collect. So I will post those data for those that weren't able to use their phone. If you don't enter your data, I mean, you still could have joined it, but all those additional questions that come from the project, if you haven't actually entered the data, then it's not going to show up. You won't be able to see that. So I'll make sure to post that for you. All right. Night, guys. Good work today.